All right. Here we have recent advances in circadian rhythms and cardiovascular systems. This paper was written by Liang Qin and Guangri Yang. It appears that both of these gentlemen have written several papers together about circadian rhythms. One of the papers from 2014 is integration of the mammalian clock and energy metabolism. They've done some with other people, timing of expression of the core clock gene. BMAL1 influences its effects on aging and survival. It's all having to do with circadian rhythm and in most cases also the cardiovascular system. Some key terms that we should go over beforehand. The circadian rhythm you're familiar with, circadian clock, CVD, cardiovascular diseases, blood pressure you're familiar with, myocardial infarction, which is a fancy word of saying heart attack, cytoplasm, which is just saying that the material within a living cell, but not including the nucleus. That's cytoplasm. All right. Abstract. Growing evidence shows that intrinsic circadian clocks are tightly related to cardiovascular functions. The diurnal changes in blood pressure and heart rate are well-known circadian rhythms. Let's talk about the diurnal, which is just saying the day and night, I believe. Daily, each day, yeah. So day and night. Endothelial function. When we talked about endo being internal, platelet aggregation and thrombus formation exhibit circadian changes as well. The onset of many cardiovascular diseases, CVDs, or events such as a myocardial infarction, heart attack, stroke, arrhythmia, and sudden cardiac death also exhibits temporal trends. Furthermore, there is strong evidence from animal models and epidemiological studies showing that disruption of circadian rhythms is a significant risk factor for many CVDs, and the intervention of CVDs may have a time-dependent effect. In this mini review, we summarized recent advances in our understanding of the relationship between circadian rhythm and cardiovascular psychology, or physiology, and diseases including blood pressure regulation and myocardial infarction. Introduction. Circadian rhythms are biological processes displaying endogenous oscillations, so internal oscillations, or cycles, of about 24 hours. These rhythms are widely observed in animals, plants, bacteria, and even cultured cells. This is Harmer and Associates, 2001. They are driven by a group of genes called clock genes. In mammals, the core clock genes consist of BMAL1, which stands for brain and muscle, aryl hydrocarbon receptor nuclear translocator like 1. So you can see why they call it BMAL1. So clock, circadian locomotor output cycles kaput, per period, and cryptochrome. They form a tightly regulated system with interlocking feedback and feed-forward loops. Figure 1. Yang et al. 2013. Which is weird. Typically you don't reference one of your own papers. That's really irregular. it's just bad form. I mean, you can if you're the only one doing the research on the stuff, but as a general rule, you don't reference yourself in a paper, and so you don't. Well, back when I did this with, that's something people do, but in science, it's not typically done. That's weird. Okay, but if they're pioneers in the field, then that would explain it. BMAL1 and clock proteins, or its paralog, NPAS2. Neuronal PAS domain protein 2 form a heterodimer, bind to E-box elements in per and cry promoter regions, and activate their transcription. Upon accumulation in the cytoplasm, per and cry proteins translocate to the nucleus, where they repress the BMAL1 clock and NPAS2. Recall, BMAL1 is the brain and muscle receptor, and the clock stands for it's a protein, circadian locomotor output cycles. I can put protein. And the NPAS2 was the neuronal PAS domain protein 2. Regulatory complex, thereby shutting down their own transcription. This core loop is interconnected with additional positive and negative regulatory loops involving nuclear receptors such as ROR alpha, RAR related orphan receptor alpha, REV-ERB alpha, which is an NR1D1 nuclear receptor subfamily 1, group D, member 1, and PPARS, peroxium proliferator activated receptors. Additionally, these clock genes control numerous target genes. Let's go back to some of this terminology in case you're getting lost. They're just receptors. That's the protein receptors. They've got fancy titles, but they're just, that's all they are. So they just receive signals via proteins and stuff. But that's all. Additionally, these clock genes control numerous target genes termed clock control genes, CCGs, thus regulating the circadian rhythms of various biochemical and physiological processes. Chen and Yang, 2014. Here they go again. Now you notice that on the other two papers I read, never did they, if they did reference a study that they had worked on before, they didn't cite themselves. Like they're like, well, we did this in this other study, but it wasn't like, you don't use citations from your own works. Like that is really bad form. And now this is the second time that they've done this. The first time was just the Yang guy, and now they're referencing a study that they both took part in 2014. I don't know, man. It really undermines my faith in this paper. I like this research paper. Like it, they shouldn't be doing that. That's a no-go. So I'll keep reading it, but I've just pretty much I would rather see somebody else's paper on this. Like this I have a very strong warning. Like I just my STEM research senses are tingling. Figure 1 is a transcriptional feedback loops of the mammalian circadian clock, and it just has the breakdown, and this figure should be fine. Like you can use it as an example, even if you don't necessarily, you take everything else with a grain of salt in this paper. It's just the breakdown of how this works, and it shows the cytoplasm and the nucleus and the receptors and the proteins and whatnot. Now they all interlocked. The circadian clock exists as the central clock in the suprachiasmatic nucleus, SCN, in the hypothalamus, and its peripheral tissue serve as the peripheral clock. The SCN, which was the suprachiasmatic nucleus, which we talked about in the first paper. I don't know if you remember. But yeah. The SCN receives light input from the retina and then conveys the photic information into neural and or humoral signals that orchestrate multifarious behavioral and biological rhythms, such as sleep, wake, hunger, body temperature, and hormone secretion cycles. Kosaka et al. 2012. Although the SCN synchronizes internal time in various tissues, growing evidence from in vitro and ex vivo experiments has proved that the peripheral clock can function autonomously without central or systemic cues. Kowalski and Brown 2007, and Takeda and Maimura 2011. Circadian clock in cardiovascular system. Circadian expression of clock genes in mouse heart, Young et al. And Aorta McNamara et al. 2001 were first described in 2001. Recently, Zhang et al. 2014 used a high temporal resolution of RNA sequence data and found that 6 to 4% of protein coding genes encode circadian rhythms and transcription in mouse heart and aorta, respectively. Ex vivo experiments displayed varied functions of mouse heart, Thurgan et al. 2007, and aorta, Keskill et al. 1996, Prosai et al. 2013, that depended on the time and tissues were collected. In addition, human hearts were found to express clock genes in a time-sensitive manner as well. Leibniz et al. 2009. Furthermore, the observations of gene cycling were extended to cultured cells. In rat cardiomyocytes, the presence of 2.5% of fetal calf serum in culture medium is sufficient to maintain rhythmic expression of core clock genes BMAL1, Rev Arba alpha, and PER2, and energy metabolic genes pyruvate dehydrogenase kinase 4, which I'm sure is just another fancy protein, and coupling protein 3, Durgan et al. 2005. Functional clocks are also expressed in cultured endothelial cells, Takada et al. 2007, and vascular smooth muscle cells, Nonaka et al. 2001. To study the role of circadian clocks in cardiovascular system, several tissue-specific clock gene deletion mouse models were recently generated. For instance, cardiomyocyte deletion of BMAL1 results in abnormal electrocardiography with prolonged RR and QRS intervals. Schroeder et al. 2013. The hearts from knockout mice were more susceptible to arrhythmia. BMAL1 deletion in endothelial cells. Westgate et al. 2008. Or vascular smooth muscle cells. Z et al. 2015 comprise the diurnal or the daily variation of blood pressure. These findings are consistent with the presence and importance of intrinsic clocks in cardiovascular system. On the other hand, although all cell types in the cardiovascular system have intact molecular clocks, these peripheral clocks need to coordinate with the central clock to synchronize responsiveness of the heart and blood vessels in diurnal variations in their environment. This disruption of normal day-night cycles, such as jet lag, leads to desynchronization between central and peripheral clocks, heterogeneity of entrainment kinetics between different organs, dysregulation of clock genes, Kiesling et al. 2010, because circadian clocks control a large number of tissue-specific CCGs, Zhang et al. 2014. The disruption of this mechanism will initiate a chain reaction to result in perturbation of a wide range of biochemical and physiological outputs, potentially contributing to the incidence of cardiovascular disease, CVDs. For example, using a mouse model of pressure overload induced cardiac hypertrophy, Martino et al. Found that rhythm disturbance by housing mice under 10 hours light, 10-hour dark conditions, adversely affected cardiac structure and function, as well as altered expression of clock genes and cardiac remodeling genes. Martino et al. 2007. Interestingly and importantly, restoration of a normal 24-hour diurnal rhythm could rescue these changes, suggesting that maintaining a normal rhythm is crucial to cardiovascular health. Day-night variations in blood pressure, BP, and heart rate, HR, are among the best-known circadian rhythms of physiology. In humans, there is a 24-hour variation in BP with a sharp rise before awakening. The highest BP value is around mid-morning. Miller, Craig et al. 1978. Let's look this word up. Because I have not heard this word before, ever. Concomitantly, at the same time or simultaneously. Many cardiovascular events, such as sudden cardiac death, myocardial infarction, and stroke, display diurnal variations within an increased incidence in the morning. Mueller et al. 1985, 1987, Elliott 1998, Rive et al. In 2013. These events, as well as kidney what the heck? Or into a couple of words here, and let's kidney albuminuria, the spilling of albumin into the urine, which may be associated with immune complex nephritis, an obsolete synonym for proteinuria. So it's saying it's an obsolete synonym for. So the more modern term for this is proteinuria. So they're using outdated terminology. They're referencing themselves, and I'm just not. Again, this paper is really sending up flags, man. In regression to end-stage renal diseases are relatively common in patients whose blood pressure fails to decline during the night, so-called non-dippers, Takata and Maimura. 2010, inverse dippers, BP rises instead of decreases at night, showed even higher cardiovascular mortality. Cario et al. 2001. These time-dependent effects are not just consequences of the sleep-wakefulness cycle or the rhythms in neuroendocrine constituents, but are also believed to be attributed to the intrinsic properties of the hearts and blood vessels whose functions show significant fluctuations during the course of the day. Durgan and Young, 2010. Paschos and Fitzgerald, 2010. Studies in genetic manipulated mice have suggested the involvement of intrinsic circadian clock and BP rhythm regulation. One of the most interesting findings is the dissociation between behavior and BP regulation. Figure 2. As the closest phylogenetic neighbor of ROR and RERB, nuclear receptor PPAR, regulates the circadian rhythms of BP and heart rate via direct interaction with BMAL1 gene. Wang et al. 2008, Yang et al. 2012. Another reference of himself. Although both vascular and global PPAR knockout mice responded to light well and displayed rhythmic behavior pattern under regular light-dark conditions, the diurnal variations of BP was dampened or even abolished in these knockout mice. This striking association between physiology and behavior strongly suggests that intrinsic clocks inside the blood vessels contribute to their functions that fluctuate in a 24-hour cycle. Several other core clock genes were also reported to regulate BP in various ways. Global deletion of BMAL1 in mice abolishes the circadian rhythm of BP, which is accompanied by hypotension likely due to the reduced production of catecholamines, Curtis et al. 2007, or the lack of BMAL1 in vascular smooth muscle cells, Z et al. 2015. By contrast, double deletion of CRY1-2 genes in mice give rise to salt-sensitive hypertension, Masuki et al. 2005. It's worthwhile to note that the intrinsic circadian regulation of BP in humans remains to be determined. Kirchhoff et al. Failed to detect a significant 24-hour variation of blood pressure in human when individuals were subjected to a 26-hour constant light condition, while the heart rate exhibited a significant circadian pattern. Kirchhoff et al. 1998. On the contrary, Scheer et al. Found that independent of environmental and behavioral changes, the endogenous circadian system modulates diurnal BP variation in humans, Scheer et al. 2010, while circadian misalignment by scheduling a recurring 28-hour day for 8 days will induce hypertension and other adverse cardiometabolic implications, Scheer et al. 2009. Conventionally, most of the hypertensive patients were treated with an anti-hypertensive medication in the mornings, based on the consideration of the day-night variation of BP. In the previous studies showing BP lowering effect of low dose of aspirin at bedtime, Hermida et al. 2003-2009. Hermida et al. Compared the potential differential reduction of CVD morbidity and mortality risk by bedtime versus upon awakening hypertension treatment, scheduling a large scale 2,156 untreated hypertensive subjects and long-term median follow-up of 5.6 years study. They found that the patients who took at least one regular antihypertensive medication at bedtime gained better BP control and exhibited a significant reduction in CVD risk. Interesting. That's not their finding. That's somebody else's findings. Hermida et al. 2010, which we were talking about, the melatonin and how it could help. They're saying that even aspirin and other antihypertensive medication at bedtime can help with your blood pressure control and reduce the risks of cardiovascular disease. So that's interesting. Although their results were impressive, independent antihypertension studies and intensive studies on hypertension-related complications are required to confirm the time-dependent effects of antihypertensive drugs and to establish chronotherapy to manage hypertension. They're saying, like, well, we need more studies on humans. And they're wanting to get like a little impressive independent and intensive studies. They're proposing some rather risky stuff, man. It's what they're wanting is to basically really mess up your system and see how how messed up it can get. But in people, that's what they're suggesting there is it's not just immoral, it's slightly unethical. Maybe not slightly might be putting it too nicely. Like it's unethical what they're I don't know. I don't know about these two guys. The more I read about, I don't know. Circadian rhythms and timing of onset and tolerance to myocardial infarction (MI) have been well established. It has been reported that the occurrence of MI is 2 or to 3 times more frequent in the morning than at night. Mueller et al. 1995, Kulik 2014. In the early morning, the increased systolic BP and HR results in an increased energy and oxygen demand by the heart, while the vascular tone of the coronary artery rises in the morning, resulting in a decreased coronary blood flow and oxygen supply. This mismatch between supply and demand elicits the high frequency of the onset of MI. In addition, plasminogen activator inhibitor 1, Kernick 1995, and many platelet surface activation markers such as GPIB and P-selection. Schneer et al. 2011 displayed a circadian pattern with high levels in the morning, which is coincident with the morning peak of thrombus formation and platelet aggregation. Toffler et al. 1987, Shearer and Shea 2014. The resulting hypercoagulability partially underlies the morning onset of MI. Disruption of circadian rhythm like shift work and jet lag has been well established to be a risk factor for many CVDs, including MI. Knudsen et al. 1999. Even a 1-hour shift, such as the transition from regular time to daylight savings time, can significantly increase the chances of MI occurring. Jansky and Lujung 2008. A series of cardiac functions related to the heart remodeling after MI are also known to have circadian variation. The early healing after MI relies on coordinated removal of necrotic tissues through an early inflammatory phase. Frangogenesis 2012. Followed by replacement and remodeling of the myocardium and extracellular matrix deposition. Lin et al. 2011. As remodeling progresses toward the maturation phase, the heart undergoes size, shape, and structure changes, which lead to ventricular dilation, dysfunction, and ultimately failure. Lin et al. 2011. Most recently, Ali Bahai et al. 2014 demonstrated that short-term disruption of diurnal rhythms after myocardial infarction adversely affected the early inflammatory phase of left ventricular modeling, altered the innate immune infiltration and scar formation, eventually led to exacerbated maladaptive cardiac remodeling in mice. In contrast, maintaining normal rhythms throughout the course of the disease better preserved cardiac structure and function. Although no animal model can completely reflect patient experience, maintenance of normal diurnal rhythms during the recovery phase after MI should still aid in a coordinated and effective infarct healing response and improve patient outcome. Moreover, clock genes may also exert non-clock roles in the cardiovascular system, which should be taken into account when interpreting the effect of circadian disruption. For instance, activation of an adenosine receptor, ADORA2B, acts via PER2 but not other clock genes to induce an energy utilization switch from fatty acid to glucose in cardiomyocytes, which promotes glycolysis and protects against cardiac ischemic injury. Eckel et al. 2012, Yang and Fitzgerald 2012. Circadian rhythms and other cardiovascular systems. Numerous animal models and human epidemiological studies have also proved the adverse effects of circadian disruption in other CVDs. Mouse hearts in rhythm-disruptive environments are prone to malfunctions with altered clock gene cycling and reduced contractility. Martino et al. 2007. Clock gene deletion or mutation in mice dampened cardiovascular circadian rhythms accompanied by dilated cardioempathy. Lefta et al. 2012. Arterial stiffness, Ina et al. 2010, or endothelial dysfunction, Diswemberin et al. 2007, Wang et al. 2008, and Ania et al. 2009. Impaired cholesterol metabolism and increased development of atherosclerosis was also verified in clock mutant mice on a Western as well as normal diet. Pet al. 2013. Aortic grafts from BMAL1 knockout mice transplanted into wild-type mice developed robust arteriosclerosis without affecting systemic hemodynamics. Chet et al. 2011. This data suggests that the intrinsic circadian clocks in blood vessels exerts significant roles as an autonomous influence in arteriologenic diseases. On the other hand, CVDs affect clock gene expression as well. For example, in salt-sensitive rats, high salt diet induced cardiac hypertrophy is associated with the attenuated rhythmic expression of core clock genes. Mori et al. 2003. Aortic constriction induced pressure overload, which decreased the amplitude of circadian expression of clock genes in the rat heart. Young et al. 2001. Durgan et al. 2005. In a type 2 diabetic rat model, cardiac clock genes exhibited a phase shift with a 3-hour delay, suggesting a loss of normal synchronization in diabetic hearts. Young et al. 2002. However, in high-fat diet-induced obese mice, vascular tissues are less sensitive to pathological disruption of circadian clocks than adipose tissue. Prosai et al. 2013. This evidence raises the possibility that although all cardiovascular cell types poses functional circadian clocks, this mechanism may be regulated in a cell type specific manner. Desynchronization between different organs, e.g. heart and aorta, or cell types, e.g. VSMCs and ECs, could occur during specific physiological and pathological situations that may give an increased chance of CVDs. Moreover, the day-night variations of blood pressure, heart rate, and baroflex sensitivity, a homeostatic mechanism for maintaining blood pressure, also coincides with diurnal variability in many other CVDs or events, such as cardiac arrhythmias, atherosclerosis, and sudden death. Portability et al. 2012, Yang et al. 2013. The timing of sudden cardiac death displayed circadian variability. It has a circadian pattern prominent in the early morning, similar to that described in patients with coronary artery disease. Miller et al. 1987. Both atrial and ventricular arrhythmias appear to exhibit circadian pattern as well, with a higher frequency during the day than at night. Portalupe et al. 2012. In hospital, many arrhythmias are observed as a consequence of MI. More complicatedly, circadian disruption not only impairs cardiovascular functions, but also been linked to other diseases such as obesity, diabetes, immune disorders, mental illness that may affect each other. Harrington, 2010. Therefore, controlling or prevent diseases that are related both to circadian rhythm and to cardiovascular functions becomes very important. Conclusion. Cardiovascular disease is the leading cause of death in many industrialized countries. Intensive effort has been made to understand the basic mechanisms. One field of investigation in recent years is the study of circadian rhythms. Increasing evidence has shown adverse effects of circadian disruption in the cardiovascular system. It becomes more and more evident and important in the modern age, particularly in developed countries, due to frequent disruptions to normal rhythms caused by shift work, artificial light, transmeridian air flight, and social activities. Bogold and Knudsen, 1999, Knudsen and Bogold, 2000. The circadian rhythms not only affect health, but also drug efficiency. It's not surprising that some drugs for treating CVDs have been reported to exhibit time-dependent effects, since there's imminent circadian function of heart and blood vessels driven by both systemic and intrinsic clocks. Although several other mechanisms outside the cardiovascular system, such as chronopharmacokinetics, Music and Fitzgerald 2013, have been suggested, the circadian clock within the heart and blood vessels should not be overlooked. Time-dependent effects should be investigated when developing new drugs for CVDs. And then there is a conflict of interest statement. The authors declared that the research was conducted in the absence of any commercial or financial relationships that could be construed as a potential conflict of interest. Well, usually I don't have to include one of those. I don't know. There's not any experiment going on here. Like, this is basically a restatement of existing data. They're not doing any experiment. There's nothing being proposed, as it were, and nothing being tested. At least they're not doing anything. They're saying it's all references to other people. So that might be okay, just because they're using other people's work and they're not doing anything themselves. Otherwise, I'd be very suspicious of this article, this research paper, because it sends up lots of warning flags. But because they're all of it's just, it's almost like a review of other people's work and then making a conclusion based on all of their work. So it's not really an original topic or an original experiment or original research. It's all based on other people's work and other people's stuff, and in some cases their own, which is kind of I had a really hard time finding anything on these two researchers as well, which already made me kind of suspicious. I don't know. This paper, I'm much more suspicious of it than the other two that we covered. But I'll let you listen to it and see what you think, and I would suggest doing some more research on these two individuals, because I had like I said, I had a really hard time finding anything on them other than other papers they had written, which is weird. Like, I even tried that site that we used to look up the other guy, and they just didn't even show up, which is odd. Some of the stuff they were proposing is really unethical. I mean, mice is fine, but like, they were wanting to do it on people, and even after they'd said that it had like detrimental effects, like life-ending detrimental effects. So it's this one is really questionable, man. But I'll let you listen to it and see what you think. All right.